Hi, in this video I will talk about enzyme coupled receptors. So we have two types from the enzyme coupled receptors so far. So the first one is the RTK, the second one is serine threonine receptor. Okay, the RTK is basically receptor tyrosine kinase. So both of these are following some kind of a similar pathway, but there are some different. They are basically if we're going to talk about molecular level, they are having different molecules and different second messengers. For the RTK, it actually goes through three pathways. And the first pathway of the RTK, the receptor, first of all, receptor tyrosine kinase. Why is it called receptor tyrosine kinase? Because in the receptor there is a tyrosine. This is an amino acid where it will be phosphorylated. So the amino acid itself, this is the part where phosphorylation will be. So when it's getting activated, a phosphate group will be bound to the tyrosine. So this is a phosphate group, and that will be tyrosine here, and this is the rest of the receptor. So also the same idea with serine threonine receptor. Phosphate group will be binding to serine and threonine. And what I have noticed about these two receptors, that we have only the amino acids that has an OH group. So tyrosine has an OH group, serine also OH, threonine also OH group. So this is what I have noticed about these amino acids. And maybe this is why they are binding to a phosphate group. They are the best binding molecules to phosphate. So as I just said, the RTK receptor tyrosine kinase, they are having three pathways. And one example of this is insulin. So insulin uses the PI3K pathway, and this is a type of a kinase. So first of all, when the blood glucose level is really high, what's gonna happen is that insulin has to be secreted, right? So, and when insulin is secreted, it enhances what? It enhances the diffusion of glucose into the cells, right? So this is the main purpose, is insulin would get the glucose into the cells. So as we can see here, this is our insulin. It goes in and binds to the RTK, which is the receptor tyrosine kinase. It binds to it, and so this part here is our tyrosine kinase, and this is the binding site. So after this binding happens, the RTK, the receptor, will be phosphorylated. Both of them are gonna phosphorylate each other, and then after this phosphorylation, it will go to the IRS, which is insulin receptor substrate, where it will activate it. After this activation, the IRS will go and bind to the SH site. So the SH site, it's an amino acid. It's a part of the amino acid where it binds to the other molecules through it. So it binds to the kinase. This is our enzyme here, the PI3 kinase. And what this enzyme does, it goes and phosphorylate PEP2 into PEP3. And then PEP3 would go to activate PKB, which is a protein kinase B. And what protein kinase B do? It will go inside of the nucleus and make the expression of the transporter GLUT4 to be increased. So there will be an increase in this particular gene expression, where it will go outside in the membrane and it will enable glucose to get inside of the cell. So this is the glucose, and this is our this is the facilitator transporter, GLUT4, and this is our cell here. So it keeps going in, and this is how glucose gets in after a meal of a high glucose meal. So this is the first pathway of RTK. So the second pathway is the PLC pathway. So it's kind of the same as the GQ pathway from the G coupled protein, the GQ. Remember, from the G coupled protein, it's kind of the same way, the same pathway. So a ligand goes into the receptor, and when it goes into the receptor, it causes the phosphorylation of the subunit of the receptor, making it activated. And after the activation, it will go and bind to the PLC, the phospholipase C. And phospholipase C, as you know from the G coupled protein, GQ, the PEP2 will be taken by the PLC, the lipase, and it will be cleaved into IP3 and DAG. For DAG, it goes through it activates PKC, which is a protein kinase, and this protein kinase would actually go inside of the nucleus and change the transcription or the gene expression. Same idea for IP3. Goes to the smooth endoplasmic reticulum, where it will increase the openness of the channel of calcium, making calcium to get out. And as you know, calcium is a second messenger. It goes through cascades until it reaches to the nucleus, and gene expression will be altered. So our third pathway in the RTK is RAS or MAPK. This is a type of kinase. So it's basically the first few steps is basically the same. So a ligand would come and bind to the receptor. It will cause phosphorylation of the subunits of the receptors, and the phosphorylated parts will go and bind, but this time it will bind here with a component called GRP, and then GRP would go and bind to SOS, and then both of them are going to activate RAS from RAS to RAS GTP. And then RAS GTP would activate MAP kinases. These are kinases are like ERK1 and ERK2, ERK1 and ERK2. And then ERK would finally go inside of the nucleus. So there has to be the purpose of the whole thing is to go inside of the nucleus and change the gene expression. Go to bind on the DNA sequence where it is the regulatory sequence. So it goes and bind to this sequence and it enhances the transcription of this part, and mRNA will be synthesized for this specific gene, and then translation will be occurring, and then the protein will be synthesized. And then lastly, the second type of the enzyme coupled receptors is serine threonine receptors. This is kind of a general, it's not really that much detailed overview of how it is. So we're having the TGF beta, which is it's acting like the ligand. It goes to the receptor that we're having, which is this serine threonine receptor. The phosphate group is added on serine and threonine, and in this case it will be activated. After the activation of the receptor, it will go and bind with SMAD, and then SMAD would form a complex, and then that complex would get into the nucleus, and it will go on the DNA regulatory sequence. So let's say this is the complex here. This is the complex, it goes and set on the regulatory sequence, and then transcription will start, leading to the high expression of the gene. So basically, all the types of the enzyme coupled receptors are for either enhancing the gene expression in a particular area or just block it. And as you can see here, so we talked about RTK, which is our first one, the receptor tyrosine kinase. We had 3 pathways in it. So we had 1 PI3K pathway, 2 PLC pathway, and the third one was RAS and MAPK. And then we talked about a general overview of serine, threonine receptor.